students, welcome back to my YouTube channel. I first thank you who has subscribed on my YouTube channel, and I thank you who is going to do it right now, because it is important. If you subscribe, you will get access to all the videos that you always upload, so that you don't miss any of these videos. So thanks for subscribing, like the video. And now this is our lesson 5 in our advanced level biology. The other time we looked at the microbodies, we came to look at the nucleus, we saw the diagram of the nucleus, functions of the parts of the nucleus, and now we are coming to look at our lesson 5, which is the cell surface membrane. So welcome to our lesson, and please, if it is your first time, click on subscribe so that you can get all the videos that are always being uploaded. Now, I think in your O level you must have looked at the cell surface membrane, the one that is known as the cell membrane. And now today we're going to look at it in details, and we come to understand how it works and everything concerned with the cell membrane. So now, the cell membrane, its major function, it is a barrier between the cell and its environment. So if it is a cell, for it acts as a barrier. It prevents the things that enter into the cell and the ones that leave the cell. So that is its major function, but it has other functions, as we shall come to see. So now, when you check on the biochemical analysis of the cell, it shows that it is made up of 45% lipid, 45% protein, and then 10% carbohydrate. That was an analysis that was carried out to find out what makes the cell surface membrane. Now, we have what we call the fluid mosaic model of the cell membrane. So the fluid mosaic model of the cell membrane, it is the one that describes how the cell membrane looks like. In your O level, you just looked at the cell membrane, but you didn't go ahead to see what comprises the cell membrane. Just looked at the cell membrane, and that is why you stopped. Now, but here we have to check the structure of the cell membrane, because we looked at very many different cell organelles. We looked at how they are made. Now we're also going to look at how the cell surface membrane is made. That is what we call the cell surface membrane, and is called the fluid mosaic model. So the fluid mosaic model of the cell membrane, it was proposed in 1972 by J.J. Singer and G.L. Nicholson. So those are the two people who proposed the cell membrane, and it was in 1972. One is called J.J. Singer and another one is called G.L. Nicholson. And now, according to them, how did they describe the cell membrane? They describe the cell membrane as a dynamic structure. So it is a dynamic structure because it keeps on changing. So dynamic means it keeps on changing with bimolecular phospholipid layer. Bimolecular means that it has two layers. It has two layers, as you can even check. It has two layers. It has the upper layer and the lower layer. So that is bimolecular layer. And it has inwardly directed hydrophobic tails. It has inwardly directed hydrophobic tails. The tails which they are talking of, they are those ones which are these ones here. These ones. So those ones are the ones which we term as the tails. And now, these tails, we are saying that they are hydrophobic. They are hydrophobic tails. And they also have outwardly directed hydrophilic heads. Hydrophilic means that they love water. The hydrophilic heads, they are outwardly, they are out. So they are out because they like water, and therefore they move out to collect water. So they are out of the cell surface membrane. While the hydrophobic tails, they are inwardly pointing. They are pointing inwards. So that is according to those two scientists, Nicholson and Singer, who proposed that the cell surface membrane is a dynamic structure, meaning that it can change, and it has bimolecular phospholipid layer. Those are the two layers which we have seen, the upper layer and the down layer, and it has inwardly directed hydrophobic tails. Hydrophobic means that they don't like water, they hate water. So those are the hydrophobic tails. Those tails are hydrophobic simply because they don't like water, and that's why they are outside. So it has hydrophobic tails and outwardly directed hydrophilic heads. The heads are outside, meaning for them they like water. They are pointing outside of the cell membrane. And they have a variety of protein molecules which have an irregular arrangements. And now these proteins, they float in lipids like islands in a sea. So those proteins which they're talking of, as you can see from this diagram here, you can see that we have some proteins here. We have the membrane protein. Now like this one is the membrane protein. We have the internal protein, the one that is called the intrinsic protein. All of those ones are some of the proteins. So for them, they are just floating on top. Now, so that is the fluid mosaic model of the cell membrane. And this one is a common question whereby they ask you to describe the fluid mosaic model of the cell membrane. So when they ask such a question, you have to first describe as it was proposed by these two scientists in 1972. Then after describing, you also draw this diagram. So from this diagram, as you can see, we have these are the heads, these ones. So those ones, they are called heads. So those heads, they are hydrophilic simply because they like water, they love water. That's why they are outside the cell membrane. We also have the hydrophobic tails. And now these hydrophobic tails which we are seeing, these are the hydrophobic tails. As you can see from the diagram, we have the hydrophobic tails, these ones here, those ones, these ones, those ones, those ones. So for the case of those ones, for them, they are inwardly directed simply because they don't like water. So that is how we describe the structure of the cell membrane, and those are the parts that make up the fluid mosaic model of the cell membrane. We have the hydrophobic tails, as I've seen, the membrane protein. Those are the proteins which are always floating in the lipids. So that is the structure of the fluid mosaic model. You can pause the video and draw. Now, having looked at the fluid mosaic model of the cell membrane, we need to also know the functions of the cell membrane. But remember, we said that the major function of the cell membrane is to act as a barrier between the cell and its environment. It acts as a barrier, therefore it prevents the things that enter within the cell and the one that leaves the cell. Now the first one on the functions is that it forms a barrier between the cell and its environment. Just as we have been saying, the first one is that it forms a barrier between the cell and its environment. Then the second thing is that it provides mechanical support and flexibility to enable the cell move, grow, and divide. So because it is outside, it is like a gate outside. So it prevents, it provides mechanical support. In case the cell is to be injured, it is done that it provides support to the cell so that it is not damaged. Let us look at another function of the cell membrane, is that it controls the passage of materials in and out of the cell, just as we have seen. It helps the cells to recognize other cells with the help of membrane protein. So it helps the cells to recognize other cells with the help of the membrane protein, which we saw from the diagram. So that membrane protein is also very, very important. It is also a receptor site for hormones and neurotransmitters. It helps in transmission of nerve impulses, the nerve impulses. So it helps in their transmission of these nerve impulses. It also helps in nerve inflation. It acts as a surface for reactions in a cell. It acts as intracellular transport system. So intracellular transport system, whereby some systems are transported within the cell with the help of the membrane protein. Sorry, with the help of the cell membrane. And lastly, it provides separate intracellular compartments, hence isolates different chemical reactions, e.g. photosynthesis in chloroplast is separate from respiration in the mitochondria, yet all the two processes take place in one cell. So now, for it has the ability to make sure that the two processes which are taking place within the cell, they take place at different times. Take an example. We have the photosynthesis that takes place in the chloroplast. And remember, chloroplast and the mitochondria, they are found in the same cell. Here we are talking of a plant cell. Now, for it is there to make sure that it separates these two processes. Photosynthesis should take place in the chloroplast, while respiration should take place in the mitochondria. So, there it provides separate intracellular compartments. So, those are the functions of the cell membrane. Now, having looked at the functions of the cell membrane, let us look at the functions of the components of the cell membrane. The cell membrane, as we have seen from the diagram, it has also the components. Now, what are the functions of these components of the cell membrane? Number one, we have the phospholipid layer, as we have seen. The phospholipid layer is the other upper and down layer. Now, what is its function? For it, it affects the fluidity and permeability of the cell membrane. In case it affects the fluidity and permeability, making it permeable to some materials to enter or impermeable. Then you also have the second part that is called cholesterol. Cholesterol, it is also in the diagram, as you saw it. From this diagram, we have the cholesterol. It is there. Cholesterol is found here. So that cholesterol, it also has a function, and it makes the membrane more fluid at low temperatures and less fluid at high temperatures. So during low temperatures, it makes sure that the cell surface membrane has more fluids, and at high temperatures it makes it less fluid. Having looked at the cholesterol, let us come to look at the glycolipid. The glycolipid, it acts as a recognition site. So if you want to test your blood group, they have to check on your glycolipid. For example, the ABO blood group system. It is as a result of different glycolipids on the cell membrane of the red blood cells. So for the doctors to determine your blood group, they have to use your glycolipid that is found on the cell membrane. So from the diagram above, you can go there and check the glycolipid is there. So it also makes the membrane more stable. It increases on the stability of the membrane. We have also seen some proteins around the cell surface membrane. Now these ones, they provide structural support to the cell membrane. They also assist in active transport of materials across the cell membrane, and they also act as recognition sites for the enzymes, energy transducers, and electron carriers. That one is clearly understood. Now, we also have the glycoproteins. Now, the glycoproteins, for them, they act as recognition sites, e.g. the neurotransmitters and the hormones. So they act as recognition sites. Now, that is all about the cell surface membrane. So, from the cell surface membrane, you have seen its function, you have seen the fluid mosaic mode of the cell membrane, how it was discovered, how you can describe the fluid mosaic mode of the cell membrane, all the functions. You have seen them, and you have seen the function of each of the components of the cell membrane. So we have finished the cell membrane as a cell organelle. So now, let us come to look at the next cell organelle, which is the vacuoles. So we are about to complete our cytology. So let us come to look at the vacuoles. From the vacuoles, we shall look at other cell organelles. Now, for the case of the vacuoles, a vacuole is a fluid-filled sac bounded by a single membrane. It is bounded by a single membrane at the end, one membrane that we always call the tonoplast, just like the nucleus, the one which you saw. Sorry, just like the vacuoles which are found in plant cells or in animal cells. They have one nucleus that is called the tonoplast. So now, in mature plant cells, there is usually one permanent and central vacuole containing a solution of mineral salts, sugars, amino acids, wastes, e.g. tannins, pigments, etc. So this one is very clear in plant cells, whereby they have one permanent central vacuole. And this one, in your O level, you could say that it contains the cell sap. So that cell sap is a solution of mineral salts, the sugars, the amino acids. We also have the wastes like the tannins, as we shall come to look at the tannins, what they do. So those are the plant cells and the vacuole. So it has one permanent cell vacuole. But when you come to animal cells, you have to be careful. For them, they are much smaller and they are many, like the food vacuoles, the phagocytic vacuoles, the contractile vacuoles. So like the contractile vacuoles, which are for osmoregulation in amoeba. So for the case of animal cells, they have many vacuoles, while a plant cell has only one large permanent cell vacuole. For the case of animal cells, they are small. Now, what is the function of these vacuoles? Are they functional? Are they important? Now, the first thing is that the sugars and amino acids in it, they may act as temporary food stores. Sorry for the mistake. So it is the temporary food stores. The sugars and amino acids in it may act as temporary food stores. Also, the wastes like the anthocyanins, they may also give attractive color to petals and fruits to attract pollinating insects and animals for dispersion. So you're always seeing different petals of different flowers. They are always having those colors. So those colors, they are due to the wastes which are found in the cell vacuoles. Those vacuoles found inside the cells are the ones which contain those wastes like the anthocyanins. These are common in plant cells, whereby they give color to the petals such that they can attract the pollinating insects, and they even change the color of a fruit such that a fruit becomes attractive, so that they can attract animals to come and disperse them away. Another function is that the vacuoles, they act as temporary stores for organic wastes such as tannins. So the vacuoles act as temporary stores for organic wastes. Those wastes like the tannins which we have seen, for them, now they are kept within the vacuoles. And also the vacuoles, they usually contain hydrolytic enzymes, therefore they perform the functions similar to those of the lysosomes. You know, lysosomes, as we looked at in our previous lessons, they contain the hydrolytic enzymes, and we saw how they act on different food substances. Therefore, these vacuoles, they contain those hydrolytic enzymes, and therefore they perform those functions which we saw that they are performed by the lysosomes. And also lastly, we are saying that the vacuoles support herbaceous plants by providing osmotic system and turgidity for support. So these vacuoles are also important in providing support to herbaceous plants and providing osmotic system and turgidity, which is for support. Now, we have looked at the vacuoles, and these vacuoles, they are very important, especially in the coloring of the petals to attract animals and also to attract the insects to pollinate flowers and also to encourage dispersion. So having seen the cell membrane, we have seen how it works, the fluid mosaic mode of the cell membrane. We We have seen the cell vacuole, the vacuoles, plant cells and animal. We have seen how they contain these vacuoles, and now in our next lesson we are going to look at the storage granules, and we are remaining with very few cell organelles so that we can finish our cytology. So thanks for watching. Subscribe if it is for your first time, and please like the video in case it has taught you something good, and please do not forget to share it to your friends so that they can also follow these lessons. Thanks for watching and have a good day.